Hello, this is top of the hour, so let's start the Center of Precision Health Research Seminar this Thursday. My name is Huan Mo. I'm the director for Cohort Analytic Core, and Dr. Rashmi Kanagal is our guest today. So Dr. Kanagal is from MD Anderson Cancer Center. She is a hematopathologist and molecular genetic pathologist and professor and director of the Microarray section for the Molecular Genetic Laboratory, and the co-director of the Bone Marrow Laboratory. Dr. Kanagal is a board-certified molecular genetic pathologist and a hematopathologist at University of Texas MD Anderson Cancer Center. She is a professor. I think I hope you mentioned that parts. And Dr. Kanagal is invited lead author for the 2022 World Health Organization classification of hematolymphoid tumors, and the author of the WHO Genetic Tumor Syndromes, and her work on the myeloid neoplasms with isochromosome 17q has been led to the recognition of this as a provisional entity in the latest international consensus classification system. And Dr. Kanagal-Shamanna is the current president of the Cancer Genomics Consortium, CGC. Actually, it's a very good conference. Highly recommended this conference. And co-leads the Cancer Genomic Clinical Genomic Clinical Resources Somatic Hematologic Malignancy Taskforce. And she served as a past chair of the Professional Association of Collaboration Committee tasked with the development of the guidelines for the appropriate utilization of genomic assay and in patient care. And there's some housekeeping for the seminar is that people from online can submit a question on the Q&A sections, and people in the room can just walk into the mic. And also is that this session is recorded, and Dr. Kanagal take over. And by the way, she is my mentor in doing my residency, actually fellowship. Thank you. Take over, please.
Thank you, Juan, for that kind introduction. As Juan said, he worked with us at MD Anderson for his hematopathology fellowship, and it makes us proud to know that he's thriving at the NIH. And so I'm giving my talk today on the optical genome mapping, which is kind of a relatively new tool for identification of chromosomal abnormalities. And we've been working with OGM, as we call it, for quite a few years now. And we've had tons of experience, and based on which we went live by implementing it as a clinical grade assay in our Department of Hematopathology. And we now offer it for every new patient that comes to MD Anderson. By that, I mean heme malignancy patient. And I wanted to kind of share with you today my experience with it. Firstly, to introduce you all to the technology per se on how it can identify all types of structure variants as well as copy number changes. My experience using OGM supplemented with the NGS for improved diagnosis, prognosis, and treatment, specifically for myelodysplastic syndrome, which I studied as a prototype and now expanded to other diseases. And finally, to kind of share with you a few case studies on how we have identified additional abnormalities using OGM during our routine testing and how it impacts therapy decisions at MD Anderson. And I have no relevant conflicts of interest. So to begin, if we kind of look at the genome, in terms of working up for heme malignancies, we've used conventional karyotyping or the G banding, conventional banding karyotype, called different ways, on pretty much every type of heme malignancies, whether it's MDS or AML, ALL, etc. And we use that to kind of risk stratify the patient. So it's kind of a very essential component for risk stratification and treatment of every type of heme malignancies. And we've been using conventional karyotype since its inception in all of the prognostic guidelines, supplemented with FISH selectively in certain scenario or other technologies like RNA-seq. And we've not really gone beyond that to identify comprehensively at a whole genome level at a super high resolution. In terms of, on the other hand, to look for mutations, we perform targeted deep coverage NGS. Pretty much every lab across the world now has at least a targeted panel NGS. And for acute myeloid leukemias and certain MDS cases, we supplement with FLT3 mutations. So it's kind of a contrast to draw. More recently, however, there are several technologies that have come through, like the long-read sequencing, whole genome analysis, to kind of look for the abnormalities, especially structure variants at a higher resolution. And optical genome mapping is one of them. It is quite distinct from the rest of the sequencing-based technology because this is a mapping technology. So what is OGM? So we all know when we look at a chromosome, when we perform the G banding, we have the dark bands and the light bands, right? So we interpret the chromosome, what number of the chromosome, the region of the deletion on the chromosome, etc., all relative to this banding, the dark and the light regions. So OGM is essentially a super high resolution banding, we could call that. It measures its genome-wide assessment. So we look at all of the chromosomes, just like the conventional karyotyping. And it's able to evaluate the changes in the sample of interest based on the fluorescence labeling. So to go into more detail, it's essentially a high-resolution karyotype. And we use DLE1 enzyme-based fluorescent tagging. And these tags are specific for a 6 base pair sequence, which is the CTTAAG. Now, this sequence is pretty ubiquitous in most of the human genome. It's in fact seen in about 15 times in every 100 kilobase strand of DNA. So using that from a sample, say bone marrow or peripheral blood, once we extract a very ultra high molecular weight DNA and kind of tag them with these fluorescent tags, and then these DNA molecules are then linearized by passing them through nanochannel arrays, which is driven by electrophoretic current. So we get these tags in a barcode pattern, essentially. So the pattern of these tags are pretty unique in different regions of the chromosome. And the same way, by comparing to a reference genome, we can tell whether alteration is seen in a particular location, genomic coordinate relative to the reference. Now, a gene can have the multiple tags with a unique pattern. It's just that the higher the number of these tags or the markers, higher the uniqueness. So certain regions inherently have a very high confidence level when we are interpreting. Now, I mentioned the ultra high molecular weight DNA because these are longer molecules compared to the routine DNA we use for sequencing-based technologies. So essentially, we are preserving much longer lengths of DNA because there are certain repetitive regions that happen to be in the human genome. In fact, a quarter of human genome has these repetitive regions that we hope we do not want to break that down because many of the structural changes happen in these areas, and we want to be able to retain that information. And it's important to note that this is a video of these DNA molecules that are passing through the nanochannels into a linear pattern. And once they come across, there is a camera that kind of captures these molecules as they pass through, and sequential images are then converted into bioinformatically into molecule data and that have these fluorescent tag pattern. Now, it's important to remember, again, there is no amplification of DNA and there's no target enrichment. So we just looking at the DNA that are ultra long. So the advantage is that we are using this ultra long DNA. So the maps, we get a longer maps that are then overlapped. There are very minimal interruptions that lead to a better confidence in the analysis of structure variants. And these are unaffected by the repetitive regions that I mentioned, where the structure variants generally happen. So the accuracy level is super high compared to others. So once we obtain the results, I'm going to go more into it. We can look at a circus plot, so we can tell if there are gains or losses of the chromosome. We can tell when there is rearrangements. We can tell when there is insertions, inversions, etc. So the circus plot data we can zoom in to get the molecule level data. So here are some examples. So the green band is the reference genome, and the blue is the patient sample or the sample of interest. And the black bars that we see are essentially the fluorescent tags. So compared to the reference here in this first image, we can tell that there is a deletion of a segment of a chromosome. Similarly, in the bottom panel, we can see compared to the green, which is the reference, there is an insertion of material that makes these tags appear distant compared to the reference. We can tell when there is a repeat array expansions, which can be very useful in constitutional disorders. We can also tell when there is tandem duplication, like partial tandem duplications that affect certain myeloid neoplasms. Then, of course, we can tell when there is a translocation. So here we can see that there are two different chromosomes and the sample of interest has segments from two different chromosomes. We can tell whether it's in cis or in trans as well, because the orientation is preserved. And we can also tell when there is an inversion, the tags are essentially flipped. We can also tell when there is copy number changes by going into a whole genome plot. And the confidence level is pretty high with this. So essentially, you're combining chromosomal microarray results for copy number changes. In addition, you're getting the balance changes as well. So if we compare the advantages and disadvantages of OGM with respect to other comparable sequencing-based assays, so compared to the short-read NGS, it's kind of a no-brainer, right? So determination of mutations is pretty accurate, but the copy number changes can only be inferred based on the changes in coverage. So there's very low sensitivity in detecting this. On the other hand, long-read NGS or even whole genome long-read sequencing, it's pretty accurate in determining the single nucleotide variants. And it has a very high confidence in determination of structure variants as well. But from our experience and with some of our collaborators, the error rates for the sequencing is pretty high. When we translate it to clinical implementation, we are yet to see a reasonable coverage to obtain in order to simultaneously detect clinically significant mutations that have to be at least at 2% variant allele frequency. And further, to obtain that coverage, it can get pretty expensive. On the other hand, this optical genome mapping, which is non-sequencing based, we are able to confidently identify structure variants at a very high sensitivity and confidence. And because of that, it is unable to detect mutations simultaneously. Now, our argument is that most of the laboratories already have very deep coverage NGS panels in place based on the advances in the last 10 years. So OGM would be a pretty good supplement when combined with the short-read NGS in identification of a high-resolution structure variants. Compared to conventional karyotyping, of course, OGM provides a very high resolution, nearly 10,000x times what conventional karyotype provides. we are able to identify, similar to conventional karyotype, a whole genome level analysis. On the other hand, it's a DNA-based assay, so we do not require any active culture of the cells. So some cases, like lymphoid neoplasms or hypocellular MDS, aplastic anemia, which often fail the cultures, we are able to tell the karyotypic abnormalities using OGM. And it can be laborious and time-consuming, conventional karyotyping. On the other hand, optical genome mapping is heavily bioinformatics-based analysis that requires expert for interpretation. Considering in this generation, where it's quite difficult to find trained technologists for a laboratory, OGM offers a particular advantage in that area. And of course, it's kind of global, right? If you need expert interpretation, it's very easy to kind of share these data files across different institutions for second opinions. So with that, I'm going to move to the next section of my talk, where I'm going to present results from a study that we performed way back in 2020, in my research lab, where we were just introduced to this technology, optical genome mapping, and we decided to kind of explore the utility. So initially, it started out as we performing OGM in about 10 samples. We didn't perform that on site. We actually shared the samples with the company. And we were, when we saw the results, we were pretty flabbergasted. So these were samples that had been stored for almost 8 years old, but we had the conventional karyotyping results and microarray results, and in OGM accurately identified all of the abnormalities and more, including some of the cryptic abnormalities that we had not picked up in these 10 cases, based on which we kind of purchased the instrument, and we decided to pursue this study in our lab in a research setting. So basically what we did was we wanted to use a cohort of myelodysplastic syndrome cases. We routinely perform conventional karyotyping as a part of clinical workup, and we would do the OGM in parallel in the research setting using the same sample. And we wanted to compare the results, like how much really did OGM results add to the existing data. The reason we chose myelodysplastic syndrome is, firstly, it's for my research interests. I work a lot on this entity. And secondly, MDS, it can be a very challenging diagnosis because it's based purely on morphologic examination of dysplastic changes that many have criticized as being very subjective because different pathologists are evaluating. I would argue otherwise. I mean, as long as you're trained, we have objective criteria to assess, but I can see why that might be a concern. And secondly, the entire treatment management of MDS is based on stratification into different risk categories, primarily based on in addition to the CBC values, the bone marrow blast percentage, it's primarily based on the cytogenetic abnormalities. So for both diagnosis and prognosis, the OGM data could be very helpful. That was our hypothesis. So as I mentioned, we started the study, we purchased the instrument, and then we were hit with COVID. So we had to kind of pause our study for several months, just because, not just because of the pandemic, but most of the visits were kind of becoming e-visits. So we were not really getting the bone marrow samples, but thankfully it resumed. We were also able to kind of use some of the stored samples for our initial validation, and it worked out okay. So we used samples from almost 101 consecutive myelodysplastic patients that presented to MD Anderson. So these were newly diagnosed, never been treated before, and the routine clinical workup was done, right? Morphologic evaluation, conventional karyotyping, and based on the results, any fish if needed for specific purposes. We also have the 81 gene targeted NGS panel that we routinely do on all patients. It's a myeloid disease focused panel. And in the research setting, we perform the optical genome mapping using the whole cells. Just a bit about our NGS panel. It's an amplicon-based NGS. Our median depth of coverage is about 3000x, and that gives the limit of detection about 2% VAF. So our results-wise, also for the optical genome mapping, our median coverage was about 300 to 350x, so about 315x. So that would again provide a LOD of about 5 to 10%. So our cohort, our baseline characteristics, as I mentioned, 101 patients showed a slight male predominance, which is kind of similar to any other tertiary center cohort. About 36% had a normal karyotype and 29% had complex karyotype. In 2 patients, we were unable to culture the cells, so the karyotype failed essentially. The distribution of the IPSS-R risk categories, which is primarily used to risk stratify these patients, is shown here. It's pretty much similar to what is seen in any tertiary care center, except that there was slightly increased predominance of patients with a higher R-IPSS based on increased P53 mutations. We kind of see that in our institution. So for interpretation of OGM, this was way back in 2020. So we have gone through several iterations of analysis. In this cohort, we probably reanalyzed all of it for 4 times before we finally came through a certain pipeline that is now available in the software. So firstly, there are multiple pipelines that can be applied. So we used a combination of rare variant pipeline, which is actually useful to identify abnormalities that are 5,000 base pairs or more with a very high LOD. So it's very sensitive in picking it up. So we can identify at a 10%, which is typical for cancer, abnormalities seen at 10% frequency in a tumor. In addition, the software has what's called the de novo pipeline, which is useful for constitutional abnormalities. So you can go as low as 500 base pairs, but it has to be present in about 40% of the tumor. So that's pretty useful for constitutional, but not really for cancer. So we used a combination of these and then settled on rare variant. In addition, in order to determine the larger copy number gains and losses, we had to supplement it with copy number pipeline, especially for those that are more than 500 kilobase pair length. Next, once we kind of apply these filters, so the size cutoff comes even before the confidence filter, right? So the confidence filter is kind of inbuilt within the software. So if a certain area of interest has only 2 fluorescent tags, it's automatically discarded. So we need to have a certain number in order to make it accurate and reliable. So all of those confidence filters have passed, and then we apply the size cutoff. And then we wanted to apply the somatic filter. As we all know, normal population, healthy population can have copy number gains and losses, as well as certain structural variants like translocations that really don't mean anything. While there are databases to identify the frequencies of copy number gains and losses in different regions of the chromosome, not many exist for structural variants. So we had to run OGM on 200 apparently healthy individuals to kind of generate a control population so that we would identify any abnormality that is not present in this population, and we would consider that as somatic. And even if it is somatic, because this was one of the first studies, we wanted to be super cautious, and we wanted to have a very high threshold in order to make the calls. So we applied what's called the clinical significance filter. So we only considered the calls which overlap either a coding region of a gene that's been implicated in a myeloid neoplasm, or any chromosomal locus that's known to be implicated in a myeloid neoplasm to be significant, clinically significant. So anything that is seen in intronic region of a known gene, we would discard it. And anything in a different gene that's never been implicated in myeloid neoplasm, based on the literature, we would not count it. So our data would have a super high level of confidence. But then there is all these others that we've not considered for this study that could potentially lead to discovery. And of course, as I mentioned, the coverage was about 315x. So that would provide an allele fraction of 10%. And if it's a heterozygous in a heterozygous population, so that's essentially 1 in every 20 cells or so, similar to the conventional karyotype. So to share with you the results, overall, we had about 4,000 plus variants by OGM in the entire cohort. About 383 of them fell into the bracket of clinically significant. We thought was pretty high confidence clinical significance. So these included not only copy number variants, but also structure variants, like inversions, several inter and intrachromosomal translocations, partial tandem duplications. Now, this was actually, before this, MDS is kind of considered to be a disease with very high frequency of copy number changes. While that was the case, the frequency of structure variants, as we determined by OGM, was not low. It's pretty high. It's just that these were not picked up by conventional karyotype. So we thought that was pretty interesting. And the partial tandem duplications, at the time, we knew that the KMT2A PTD was a bad prognostic marker seen in AML. And we also identified in our cohort about 3%, 3 cases or so, which kind of fell into the clinical significance bucket. And as I mentioned before, we also kind of experimented with the de novo pipeline. So we wanted to determine the copy neutral LOH using those probes. The limitation is that at this time, the OGM can identify based on the SNPs, the CNLOH, but it's not that sensitive. So only SNPs, only CNLOH that's more than 12 megabases, and it has to be seen in a majority of the tumor population with at least 40% VAF. So that's the reason we did see CNLOH, but it was misleadingly low. But this is, of course, not accurate. Here is an example of some of the abnormalities that we see here. Here's the circus plot showing copy number gain, loss, other deletions and insertions, as well as translocations involving 1-7. And here is the deletions, insertions, tandem duplications, inversions. Oh, I didn't hear. Was there a question? Okay. And so in addition, we can see that the cohort included translocations. There were cis translocations and intrans as well, where the other gene is flipped around. Using the circus plot, we are able to infer 3-way rearrangements as well. We can see this is a different case where there are multiple translocations, so-called the chromothripsis or chromoanagenesis patterns. So interestingly, this case by conventional karyotype just showed 2 abnormalities, and it was in composite setting, but the OGM kind of unveiled this whole chromothriptic event. So how many of these, our next step was to compare these abnormalities with our conventional karyotyping that were performed in our routine clinical workflow. And a staggering number, almost 58% of these abnormalities were not detected by karyotyping. Now, we already know, right, the technology is a high resolution technology, so it makes sense. These were actually the 58% of abnormalities were seen in a third of the population, 1/3 of our cohort. And these were either submicroscopic or they were not detected because the karyotype was seen in the setting of poor morphology. The more complex the disease, the more messed up the chromosomes are, and the morphology is pretty poor. So the sensitivity in detection of these abnormalities using traditional techniques also drops. And then some are, of course, routinely cryptic. For example, the NUP98 translocation or KMT2A, certain MECOM rearrangements, they can be very hard to detect using conventional techniques. So when we compared between these two technologies, so we found that there was 97% concordance, which we thought was pretty reasonable. And in addition, the OGM, because it's a DNA-based assay, it can pinpoint all of the ambiguity that's associated with it. So any of the markers or additional materials, sometimes the material that was inserted, we were able to actually specify which genomic region that was. I also mentioned 2 patients where that had insufficient metaphases. And we were able to get informative results in both of these patients using OGM, and they did show abnormalities. So it kind of improved, it could potentially improve the risk stratification, accuracy of the risk stratification if we use OGM data. And the overall frequency of normal karyotype, which was about 36%, dropped by 16% because we were able to uncover underlying abnormalities. Even in cases that already had genomic abnormalities, OGM showed a higher degree of genomic complexity compared to conventional. So some of the examples, so in order to kind of assess how it clinically impacts, we kind of narrowed down on certain markers. So the KMT2A rearrangements, deletions, gains, and PTDs. For TP53 deletions, MECOM rearrangements, NUP98, deletions affecting TET2, ETV6, we know these make a difference in terms of how, if we do see changes in this, the management changes potentially. So we focused on those. As you can see here, if you look at KMT2A alone, the OGM was able to identify PTD in all 3 cases. And of course, it's cryptic by karyotype, so that's a given. One thing I forgot to mention, that because it's one of the first studies, all of the changes that we saw by OGM here, we confirmed new changes seen by OGM, not seen by conventional karyotype. We confirmed it using an orthogonal assay, whether it's a FISH or a PCR or MLPA. We use a combination of techniques. And in terms of MECOM rearrangements, almost 80%, 5 cases had MECOM, and only 1 case was evident by conventional karyotype showing some sort of abnormality confirmed by FISH. In 4 cases were completely missed, so almost 80% detected by OGM alone. NUP98, there were 2 cases, and 1 was uncovered by OGM retrospectively, and TP53 alterations, and others as well. So we can see that OGM is able to identify clinically informative changes in MDS in this particular study. As I already mentioned, the OGM doesn't provide NGS studies, so the data that we have from OGM is completely complementary to the NGS. So as I mentioned, for diagnosis of myelodysplastic syndrome, it always helps to have some sort of a clonal marker, right? These, by conventional cytogenetics, about 50% of cases have a clonal marker. And if you use NGS, about 90% of the cases have a clonal abnormality. But by using OGM and NGS, almost 98 of the 101 patients that were diagnosed as MDS did have a clonal abnormality, which is pretty significant. Now, the next question is, so what does that mean? Yes, OGM can identify these abnormalities at a higher resolution. Does it really make a difference for the patient? So for that, I wanted to talk a little bit about the IPSSR stratification. So in order to risk stratify these patients, so every MDS patient is risk stratified in the clinic, and depending on which category he or she falls into, whether it's very low, whether it's very low, low, intermediate, high, or very high, the management differs. And how we determine this is based on multiple factors, right? One is the cytogenetic risk group, and then, of course, the bone marrow blast and the CBC values. Now, the cytogenetic risks group, if you go up here, depends on the type of the abnormality, the number of abnormalities, and whether this abnormality is seen as independent clones or whether it's a part of the same clone, etc. So it's pretty, cytogenetics plays a super important role in this. So this scoring system is called comprehensive cytogenetic scoring system, based on which you assign a score to add to the rest of the parameters to get the final risk group. So we use the CCSS based on our conventional karyotyping, and we also applied the same tool using the OGM data. And it did, using the OGM data, of course, the CCSS score changed in almost 21% of the patient. And by applying that to the RIPSS, 17% of the patients changed the risk category. So you might say it's a high resolution. Yes, you'll get a higher worse score because you're able to identify more abnormalities. But what is shown here below is actually crisscross, because in some patients, there was downgrading as well. We uncovered certain abnormalities like -Y or deletion of the 11 chromosomes 11q, which is actually favorable. So it downgraded some of these patients. And in some patients, we were able to tell the two different abnormalities were actually a single alteration resulting due to a rearrangement. So that counts as one abnormality, which also downgraded in certain patients. And the three-way abnormalities just counts as one, right? So there was downgrading as well. So we, even though this was a very small group of patients, about 101 with a fairly short follow-up of about 3 years, we did correlate with the outcome. And it turned out that the OGM prognostication had a higher correlation with the outcome compared to the CCSS. But this was a single institution study, short follow-up. So this needs to be confirmed or validated by other institutions as well. But that's kind of making a headway into potentially better prognostication scores moving forward. The other thing is the actionable variants, right? So what are those? For example, in our institution, we have the menin inhibitor trial, which is pretty successful. The preliminary studies are showing that to be very promising. So uncovering abnormalities affecting MECOM or KMT2A, especially rearrangements, are actionable because the patient could potentially become eligible for this trial. So if we count them as actionable variants, was actually seen in 15% of the patients. So based on this study, this particular study, we concluded that the unbiased whole genome high-resolution structure variant profiling using optical genome mapping revealed a higher frequency of structure variants in MDS than what was known at the time. And these are clinically significant SVs. It can be seen in nearly a third of all MDS, and that's being super conservative. And these findings are complementary to targeted NGS. So using a combination of OGM and ultra deep coverage NGS in a clinical setting could probably lead to uncovering newer abnormalities that could be potentially, that could improve or provide data for future precision medicine. In applying directly to diagnosis, we know, I just wanted to give you how it kind of helps the lab in terms of workflow as well. So the latest WHO and ICC classification of different hematologic malignancies kind of heavily incorporate genomics into it, right? Not just mutations, but majority are rearrangements. For example, in myelodysplastic syndrome, the presence of any of these abnormalities within the box it implies that it's not MDS anymore. The diagnosis is upgraded to AML, regardless of the blast count, which is a big change compared to the previous classification system. And I already mentioned to you about the implications of identifying certain abnormalities for prognosis and treatment selection. And in terms of AML, the subclassification based on the WHO is kind of shown in this slide. So if you can imagine, some of these, again, are cryptic, right? Many of those involving the KMT2A rearrangements and MECOM rearrangements can be quite cryptic. So in a clinical lab, if we were to get a case of AML, it's almost impossible to kind of routinely perform all of these FISH tests in cascade to determine the accurate category where it belongs to. So having a genome-wide assay, a quick implementable clinical assay would be super helpful for accurate classification and to identify therapeutics, same menin inhibitor trial. And similarly, in B lymphoblastic lymphoma, this is where it's extremely helpful, right? So many of these, again, are driven by cytogenetic abnormalities. And the one highlighted here, the BCR-ABL1-like feature, we still don't know what abnormalities affecting ABL1, ABL2, JAK2, all of those fall into this category that ultimately leads to the BCR-ABL-like profile, transcriptomic profile. And currently, the way to interrogate is, of course, RNA-seq, which may not be feasible in medium complexity labs. However, using OGM, we are able to uncover all the abnormalities without having to do panels and panels of FISH. So that's where it's particularly useful. This is an example of all the FISH assays that's kind of matched with the treatment that could potentially cure the patient. And in order to cover all of these, it would be almost unfathomable for a routine clinical lab. Again, for T-ALL in a similar way, now BCL11B rearrangement is extremely important for the treatment with right therapy. There are several provisional entities also shown in the ICC classification that would require tons of FISH assays or an RNA-seq. And OGM, being a DNA-based assay, is quickly implementable in a clinical lab and can be extremely helpful to appropriately apply these categories. So based on this, we and our data in MDS, we decided to validate OGM as a clinical assay in our lab. And we were able to go live in 2023. So it's been more than 1.5 years since we've gone live, and we perform OGM routinely on all patients along with conventional karyotype. We haven't let go of that because most of the prognostic models use data from conventional karyotype, so we want to be able to compare that. But the results have been pretty impressive, and we are actually in the process of getting a second instrument to kind of meet the needs right now. And I wanted to share in the last few minutes, 10 minutes, some of the cases where OGM did play an important role. So here is one scenario. This was a 28-year-old man, again diagnosed with MDS 16% blast. This was a post-cytotoxic therapy or a therapy-related MDS, previously treated for Ewing sarcoma. Karyotyping showed deletion 7. It is post-transplant, so it was a donor karyotype. The FISH panel was negative. NGS studies showed mutations in RUNX1, SF3B1, and BCORL1, and there was an FLT3 TKD. But OGM, when we performed, revealed a cryptic MECOM rearrangement with CDK6. So because you can identify the genes, you're able to tell the partner as well using OGM. it's a cryptic 3-7 translocation, but the karyotype was just deletion 7q, right? So we also see the deletion 7q here. This was important because the patient was in the tumor board. The oncologist did consider treating with menin inhibitor, but did not qualify, but at least there was an opportunity to discuss that. And of course, even though it was 16% blast, the diagnosis is AML now, based on the WHO. The case 2, this was a 68-year-old patient, newly diagnosed MDS, 5% blasts, FLT3 ITD positive, normal karyotype, WT1 mutations. That kind of gives you a clue that there might be an underlying NUP98, right? The WT1 mutations with FLT3 ITD. Generally in pediatric, but in this case, he's a older gentleman. And as expected, the OGM was the one that revealed NUP98 rearrangement. And as I highlighted before, presence of MECOM or NUP98 in this case is actually AMLs. So we went back and we looked at our original cohort and we reclassified them using the WHO 2022 based on the OGM data, and about 7 out of the 101 patients actually met criteria for AML based on abnormality like genetic abnormality like MECOM or KMT2A, etc. The other important thing in MDS is the identification of monoallelic versus multiallelic TP53 alterations. So Dr. Papa Manuel from MSKCC showed that only patients with multi-hit TP53 alterations, for example, more than one P53 mutation or mutation with a concurrent P53 deletion or a copy neutral LOH of TP53 had worse prognosis, significantly worse prognosis. On the other hand, patients with a single TP53 alterations did similar to the wild type. And this has also been adopted by the WHO and the ICC classification. So it has to be MDS with biallelic TP53 inactivation. So even the, so to illustrate that, I'm going to show you 2 examples here. So this was one case, again, an MDS, 15% blast with a single P53 mutation, 37% VAF. So the conventional karyotype showed a few markers, composite karyotype, but we did not particularly see any 17p alteration. But considering that it's a complex karyotype, a high-grade MDS, we did perform FISH, and it confirmed a 17p deletion. So we can tell it's biallelic TP53. But we also performed OGM. So what else did OGM provide? So here we can see that there is, it not only shows the complexity, but the pattern shown here between chromosomes 21 and 17 kind of highlights the chromothripsis or chromoanagenesis, right? We all know complex karyotype is one thing that with time, there is accumulations of alterations, as opposed to chromoanagenesis, which is a single catastrophic event affecting a 1 or 2 chromosomes, and these segments are quickly stitched back together. And cases with chromoanagenesis particularly have a poor prognosis, even compared to those with complex karyotype. So in this case, if I have to choose between FISH and OGM, obviously OGM is much more informative. In addition to providing the 17p deletion, it can provide a whole genome view of the degree of complexity. Here is another case, again, a high-grade MDS, showing multiple P53 alterations. So we already know it's a TP53 biallelic inactivation. And what else did OGM add? I mean, it has to, because I'm using this as an example. So here is a circus plot. As you can see, there are several abnormalities, but what we notice is multiple translocations between chromosome 1 and 11. And there is this gain of chromosome 11, a lot of gains on chromosome 11. So when we zoom in, we can actually uncover this to be affecting the KMT2A. There is multiple tandem duplication of exons 6 through 8. So this is the classic KMT2A partial tandem duplication. Why is it important? So this is the molecular IPSS. So the IPSS revised, our IPSS that I mentioned before has now been upgraded to use the somatic mutation data to molecular IPSS. So if we look at the molecular IPSS, there are certain abnormalities that carry a very heavy prognostic value that are put into the score. So the top one is, of course, the TP53 multi-hit. In this case, we know there were multiple TP53 mutations, so that counts. And the second thing we see here is the MLL-PTD or the KMT2A-PTD. So the identification of KMT2A-PTD would increase the risk score, so it's clinically significant here. The patient ultimately underwent transplant. So the KMT2A abnormality, the only way to kind of detect it is using a very sophisticated NGS, which we do not have. The other alternative would be an MLPA assay. We don't have it in-house and we again need to kind of validate it. But having an optical genome mapping, it's just a single assay. We are able to tell all of these abnormalities and kind of inform the oncologists so they can be as informative as possible in order to risk stratify the patient. So in addition to PTD, in our cohort itself, there were several other alterations. All types of alterations can be picked up. So here is an example of KMT2A deletion, gain, fusion. There was a case of KMT2A with ELL gene and partial tandem duplication. So it's a pretty comprehensive assay. And finally, this case is pretty interesting, I thought. So here, this was a 74-year-old man, again, therapy-related MDS, complex karyotype. The NGS did not show any P53 mutation. So currently, the WHO and the ICC, to identify MDS with biallelic inactivation, or for now, you need to have a TP53 mutation and a deletion or a copy neutral LOH, a second alteration, or it could be more than one TP53 mutation. But in this case is interesting because there was no mutation, but the OGM data was quite interesting. We can see that there is a deletion here, 17P, that includes the P53 area. In addition, there is a rearrangement between 17, that carries the P53, and chromosome 20, that disrupts the P53. So we believe that there is indeed biallelic TP53 disruption, not necessarily through mutation. So this could potentially be a biallelic P53 inactivation, right? That we are not considering at all in any of the current criteria. We also performed a P53 IHC for this just out of curiosity. And we see that it's completely negative. Normally, you should see some weak staining, which we don't see here. That kind of suggests an inactivated profile, which we believe it's now kind of a discovery. Like, should these cases be considered in the same category and treated accordingly? So it's all something, I guess, more data, as more data accumulates, all of these open up additional opportunities for precision medicine. We were also, this is a poster we presented at ASH, where we compared the MDS and AML transformation, and we were able to demonstrate the role of chromoanagenesis involved in this transformation. And again, before I wrap up the talk, I also wanted to say that even though I focused heavily on MDS, the applications go beyond for all heme malignancies, right? Acute myeloid leukemia, it's a given. I already showed you the different sub-classification where we are able to tell multiple rearrangements of PML, RARA, inversion 16, all of the actionable alterations. In addition, genomic workup of T-ALL. This is particularly important because the lymphoblastic leukemias do not grow well, and karyotype is often normal, and most of these abnormalities are cryptic. So here is an example of a case where we found the TLX3 BCL11B fusion along with the CDKN2A deletion. a very reasonable alternative for an RNA-seq assay. So a lab, it's much easier to kind of play with a DNA-based assay compared to RNA-based. In some cases, RNA-based assay may not deliver. For example, MECOM rearrangements it's because there is no chimeric product. It's very hard to evaluate that using RNA-seq, where OGM has an advantage. So overall, the utility of OGM is pretty heavy at this stage. So in terms of looking at a pathologic workup, morphology by microscopy, essential. NGS mutation, absolutely essential. And so far, it's been cytogenetic using conventional karyotype, FISH, chromosomal microarray. But I think routine structural profiling using OGM is extremely important and relevant in this era. And finally, I already talked about this. Finally, one thing I wanted to say is the turnaround time for our, since we went live, we are able to provide a TAT of about 5 to 7 days, which is very reasonable for the malignancies while we are waiting for other results from NGS, etc. So we are the oncologists essentially are waiting for OGM results in addition to the NGS in order to come up with a management plan for the patient. And finally, we have for any other labs who are planning to implement OGM in their clinical labs, we have published through the International Consortium framework. All the authors here, we are all like early adopters. We've gone through several iterations, repeated multiple things, and we've come up with a set of guidelines to kind of ease the validation process because it's kind of a completely different technology for cytogenetic labs. And just to highlight the global impact of the OGM, the ISCN, which is the standard nomenclature guidelines for cytogenetic results, have already published the ISCN nomenclature for OGM, just highlighting that this emerging technique is here to stay for clinical workup. So with that, I just wanted to acknowledge several folks who have been involved in making this study a success in implementing OGM into our clinical laboratory. I wanted to thank Department of Leukemia, Dr. Garcia Monero, who was the first to listen to my idea and kind of supported it so I could move forward with it. Hui Yang, who was heavily involved in the bench work in terms of when it first began, several iterations after, now we have a simpler workflow. Our Department of Hematopathology, Dr. Medeiros, who was not only supportive for my research, but also during implementation of the assay and currently in our routine testing. And the BioNano team during the initial troubleshooting stages were extremely supportive of our studies. And all of my master's students here who were involved in one way or the other with this project for their help in validating some of the results and for my funding sources. And with that, I wanted to, I'm ready for any questions if you all have any.
Thank you so much, Dr. Kanagal. Do you have questions in the room? I haven't seen anything online yet. Meanwhile, I do have a question that since we have a group of germline experts here doing genetics, and this technology seems open a new vision that we have a world that you've never seen before, like the structure variants. So in that 200 healthy controls you are doing, and a lot of like filter out variants, like might not be important as hematology diseases, but do they have, what kind of those variants have you observed that since a lot of people here maybe find out they may be associated with other diseases?
So that, although I didn't focus on constitutional disorders or germline predisposition, OGM is kind of perfectly suited. It's in fact better suited for constitutional disorders, I should say. As I mentioned, there is de novo pipeline that can go down and pick up alterations as low as 500 base pairs. And there are several published literature that where the OGM, despite doing the whole exome and the whole genome analysis, OGM was able to identify something that the sequencing-based technologies have missed, both in the neurodevelopment disorders. I think it's all available in PubMed. In addition, in our cohort per se, there is an interesting case where there is, of course, it has the RUNX1 deletion affecting a few exons, which we believe to be a germline predisposition, and we believe that it's probably present in the germline. But unfortunately, we didn't have the patient for follow-up in order to perform a skin fibroblast culture. But OGM it's an absolutely great point. It's actually a very, very useful tool to uncover a germline or constitutional abnormality, especially the repetitive disorders. There are several studies that have already shown OGM replacing the southern blot and other assays.
Thank you. Any other questions here? If no, I might have another question then. So for the myeloid neoplasm with TP53 mutation or inactivation, only thing we know about them like a couple years ago was they are very bad. And the second is they're very messy. And then there's like 3 lines of karyotype. And now we have actually this kind of resolution, look at what has changed. I mean, it has a chromosomal instability. What has changed? That is anything now we know the more granular level that can be actually targetable. I know that the case you present, you have the KMT2A PDT. I'm not sure if that still can be used manually to target, menin inhibitor to target. But is there anything that come up now we know that we might be able to target for this kind of population?
For the P53 mutated cases?
Yeah.
Yeah, so the APR-246 was one of the drugs that was used, but unfortunately, I don't think it showed much difference. There are several other trials that are happening in P53 mutated cases, but it's a very difficult disease to target, and I don't know of any trial that is particularly promising. The macrophage, the CD48 target, also, I think the trial was stopped. So yeah, I mean, it's a very difficult, I think the data from OGM and additional sequencing studies might throw some light ultimately in this unfortunate population that could potentially help. And again, even in terms of mutation, not every mutation is the same. I think we've published one paper where certain P53 mutations, depending on what we call the evolutionary action score, did worse compared to certain others, even though they were both likely pathogenic or pathogenic. And I guess more data needs to come out in order to dissect this population further.
Thank you. And there's one online question. Can you see it? It's on the Q&A. It's great work, revolutionary. My question is how this technology might differentiate current knowledge and targeted therapies on established entities like SF3B1 MDS entities.
So that's a very interesting question. On established entities, like if we take the example of SF3B1 MDS entity, we know that SF3B1 mutation alone versus SF3B1 mutation with, say, del5Q or RUNX1 mutation or other alterations, the prognosis is different, at least for now. In addition, some of the SF3B1 mutated cases, at least in our cohort, did acquire MECOM rearrangement at some point. One of it was actually cryptic. So I think just in terms of diagnostics, uncovering comprehensively all of the underlying abnormalities in this disease would be helpful. And I know the SF3B1 mutated cases can have R loops that kind of can affect the response to drugs. The OGM does have the potential to dissect what we call the HRD score, which is kind of based off of these R loops. So it's something to investigate. I personally have not, but that's very interesting.
Thank you. Actually, we have Fanconi experts here. That is like an expert in the HRD.
Wow, perfect.
Thank you. Do you want to come to the mic, please?
Some solid tumors, P53 and chromosomal abnormalities kind of go hand in hand. So in some of the solid tumors associated with, for instance, Fanconi anemia, because of genomic instability, P53 and chromosomal abnormalities go more or less hand in hand, because it's kind of maybe indication of instability. Is that true in other myeloid neoplasms?
I could not, I'm not able to hear through that mic as well as, can Juan repeat the question?
So you're talking about specifically treating P53, like a answer to your question, carriers of P53 mutations. But P53 appears to be in some cases as a part of instability where chromosomal abnormalities are also associated with them. So it may be much more complex than just P53 variants alone. But maybe in hematological malignancies, there may be some only P53 are seen or chromosomal abnormalities. And I was wondering how specific the variation is in compared to solid tumors.
So P53 is mutated cases generally in heme malignancies tend to associate with del5q, chromosome 5 and chromosome 7 abnormalities and 17p. So because all of these has been assessed using conventional karyotyping, right? Any 3 or more abnormalities are considered complex and with P53 mutations are known to do bad. But I think with OGM, we will probably in the future dissect at a much better level different subgroups, even in TP53 mutated cases. And maybe there are cases with 3 to 5 abnormalities may do better than 5 to 10. You know, all of the, right now the data is not there yet, but once multiple centers start generating the data, we may be able to further develop prognostic subgroups even within those categories. And why the P53 affects certain chromosomes preferentially in heme malignancies, we don't know that either. So I think this technology may throw more light to uncover that. As of now, I don't know the answer.
Thank you.
You can do the same thing for assay efficiency and you probably have the same observations that we can. Any other questions? Otherwise, thank you so much. Thank you so much, Dr. Kanagal.
Thank you so much. It was a pleasure being here, and thank you everyone for your attention, and have a good afternoon.